everyone, I'm Sailee and I hope you all are doing well. So again, I have a new fresh lecture for you today. So before starting with our lecture, please do follow us on our WhatsApp and Telegram groups, the link of which is given in the description box of this video. And also in the description box, you will get link to our free PDF notes. All subject notes are mentioned in that link. So do click on that link to avail your notes. And also for more such wonderful lectures, doubt sections, quizzes and your free PDF notes, please do subscribe to our channel, that is EduPhy India. So let's start with our today's topic of discussion will be animal kingdom. It is going to be an interesting topic. So let's begin. So first we'll get introduced with our today's topic, that is animal kingdom. So as we all know that millions of species of animals have been described till now, and this is the reason why it is important to classify the animals so that they can be assigned in a systematic position. So in our today's lecture, we will be discussing about some basis of classification that are used to classify animals. So animals are classified on the basis of arrangement of cells, their body symmetry, nature of Selum, pattern of digestive or circulatory and reproductive system. This chapter will be divided into series. So in the upcoming lectures, we will be discussing in more details about the phylums, classes that are classified under animal kingdom. So today's our topic of discussion will be firstly the basis of classification, what criteria are used to classify animals. So let's start. First base of classification is levels of organization. So as you can see on your screen, the picture gives you an idea about levels of organization that are used in classification of animals. First level of organization is at cellular level, second is at tissue level, third is at organ level, and fourth is organ system level. So first one by one we will see what this levels of organization means. So cellular level. Cellular level, cells are arranged as loose cell aggregates. Cell aggregates means cells are collected together, that is what is called as cell aggregates. An example of which can be seen in sponges. Then in tissue level, the cells performing the same function are arranged into tissues. An example of this type of level can be seen in coelenterics. Then fourth one is organ level. Tissues are grouped together to form organs, each specialized for a particular function. And example of this level can be seen in platyhelminthes. Now fourth one is organ system level. Organs are associated to form functional systems like annelids, arthropods, molluscs, echinoderms and chordates. So in this organ system level, we will be seeing example of two systems. For example, the digestive system in platyhelminthes has only single opening, that is one opening to the outside of the body that function as both mouth and anus, and that's why it is called as incomplete digestive system. Whereas in complete digestive system there are two openings present, that is mouth and anus. Now similarly in circulatory system has two types, one is open type and second is closed type. In the open type, blood is pumped out through heart. It is not restricted or limited to blood vessels. Cells and tissues are like directly taking bath in it. And in closed types, blood is circulated through blood vessels, that is arteries, veins and capillaries. So this was about our first basis of classification used in animal kingdom, that was levels of organization. Now second basis of classification is symmetry. So as you can see on your screen, there are three types of symmetries that can be observed in animal. Firstly, bilateral symmetry. Only one plane can divide the organism into two identical left and right halves. From the image, you can have the idea that the plane that is dividing the organism, the red line that is dividing the organism into two identical halves. That is one will be the left part and other will be the right part. This type of symmetry can be observed in annelids and arthropods. The next is our radial symmetry. So any plane passing through central axis can divide organism into identical halves. Example of this type of symmetry can be observed in coelenterics and echinoderms. As you can see on your screen, the many planes are passing, the red lines, there are many red lines passing through this organism. This is an example of aquatic animal that you can see on your screen. So this number of planes that are passing through the central axis of this aquatic animal divides this organism into identical halves. Now third is the animals that do not show any symmetry, that is no symmetry, means asymmetrical. Any plane passing through median or central axis cannot divide this organism into equal halves. So this type of example, that is asymmetrical or no symmetry, can be seen in sponges. Now third basis of classification is diploblastic and triploblastic organization. In this we will be studying about germinal layers. So as you can see on your screen, the picture gives you a brighter idea. Different layers in this picture are represented by different colors for your better understanding. So let's start. First we'll discuss about our diploblastic organization. Cells are arranged in two embryonic layers. That is, one is external layer, which is called as ectoderm. The green line represents that layer. And then the another one is internal layer, that is called as endoderm. The blue line represents the layer in this picture. So there can maybe also mesoglea. Mesoglea may be present. Mesoglea is like gelatinous, means jelly-like material that is present between this external layer, that is ectoderm, and internal layer, that is endoderm. As you can see on your screen, the gray color that represents our mesoglea layer. So this type of organization, that is diploblastic, can be seen in porifers and coelenterids. Now our next organization is triploblastic organization. So as the name suggests, triploblastic means three layers are present. So three layers present in developing embryo, that is ectoderm, mesoderm and endoderm. That is why it is called as triploblastic. In the picture you can see that each layer is represented by the different color in the picture. So this type of organization, that is triploblastic organization, can be seen in Platyhelminthes to chordates phylum. Now our fourth level of classification is Selum. Selum means body cavity which is lined by mesoderm. So as you can see on your screen, the animals on the basis of their Selum pattern can be divided into three groups. First one is acoelomates, means body cavity is absent in these animals. They do not have Selum, and this pattern can be seen in Platyhelminthes or Porifera or coelenterate phylums. Then next is pseudocoelomates. Pseudocoelomates means no true coelom is there, as mesoderm is present in scattered pouches between ectoderm and endoderm, and this type of pattern can be seen in Helminthes. Now last is true coelom, that is coelomates that have means animals that have coelom. An example of this type of pattern can be seen in annelids, arthropods, molluscs, echinoderms, chordates and etc. So further about this pattern of coelom, here I have a picture of earthworm for you, as the picture is colorful, of course representing the respective layers. So in this you can see the white part, that is the cavity, which is called as coelom, which is surrounded by or which is linked by mesoderm, the blue layer, and then our last red layer is ectoderm, and endoderm is in yellow color, the yellow layer in the center, that is endoderm. So this picture gives you a good idea about the different layers that we can observe in the organisms after we segment it. So this is an example of earthworm. So our next basis of classification is segmentation. In this we will be taking again example of earthworm. As you can see in the image, the label represents the segments of this earthworm. So firstly we will talk about metamerism. Metamerism means if a body is externally and internally divided into segments, that is called as metamers, with serial repetition of at least some organs, then this phenomenon is called metamerism. And one good example of this metamerism can be seen in earthworm. So there is true metamerism, means that can be found in Annelida, Arthropoda and Chordata phylum. Annelids, segmentation is externally as well as internally. And in arthropods, segmentation is only externally, and segmentation is internally in chordates. Also adding to it, pseudometamerism that can be found in tapeworm. So the segments of tapeworm are budded off from nape. They are not embryonic in origin. So this type of metamerism, that is pseudometamerism, can be seen in tapeworm. So moving to our last basis of classification, that is notochord. Notochord means it is like a rod-like structure formed during embryonic development on the dorsal side. Dorsal side, that is back of an animal. As you can see on the picture, the label represents the notochord. It is like a rod-like structure, and it is derived from mesoderm. An example of which can be seen in chordates. All the animals that have notochord are referred to as chordates, and the animals that do not have notochord are called as non-chordates. Example is Porifera to Echinoderms phylum. So this was all about our today's lecture, that is basis of classification. In the upcoming lectures, we will be discussing more about animal kingdom, classification of animal kingdom, different phylums and classes that are classified under this kingdom with the help of examples. We will see it in our upcoming series of these lectures. So this was all about our today's lecture. For more such video lectures, free PDF notes, doubt sections, quizzes, please do subscribe to our channel, that is EduPhy India, and also follow us on our WhatsApp and Telegram groups. The link is given in the description box of this video. So thank you for your attention.